Today we will discuss the DNA preparation because in the last 2 days we discussed the cloning, the first step of the cloning procedure, including the cloning vector, expression vector, and and also the process how to introduce the DNA into the cell. That's the transformation. All right, that's yesterday. And after the transformation, then we use the selective media to identify the recombinant cell. All right, and those recombinant cell that basically the will prove the recombinant phenotype, and then to identify the recombinant genotype, then you will need to purify the DNA. That to purify the plasmid DNA, and then to prove that the cell uh, carry the recombinant molecule. All right, and then so that's why today we will discuss the DNA preparation. So to prepare the DNA, all right, for the those bacteria, that if you use the bacteria, the E. coli based vector, then you inoculate, you transform the plasmid DNA into a bacteria. If you use the phage vector, then you are transformed into the phage. Then you have different procedure to purify the DNA. Then for the bacteria cell, so basically you need to grow the cell and then harvest the bacteria culture, and then prepare the cell extract and purify the DNA from the cell extract, and then you can concentrate the DNA sample. And for phage, then you grow and harvest because you use the phage to infect the bacteria so that they can replicate quickly. So you grow and harvest the culture, and then you separate the phage from the bacteria, and then you can isolate the DNA from those phage particle suspension. So this is the general procedure that if you grow the bacteria culture, because first of all you need to multiply a lot of the cell, all right, and then that normally it's overnight, and then apply the centrifugation to collect the cell pellet, and then you collect the cell pellet and break the cell to release the cell extract, and from the cell extract, then you isolate the DNA. Okay, so that would be the general procedure of the DNA isolation. And in the first part of this DNA preparation, this procedure will be available. That generally available to purify all the DNA in the cell. And the second part, we will just focus on how to isolate the plasmid DNA from the genomic DNA. Okay, and then the first step that you need to grow the cell culture. All right, to grow the cell culture, then you use different kind of media. All right, that's from yesterday's lobster that you practice. Okay, so that to grow the media, then you have to define the median, and for example, the M9 median, so you know all the component. So that's the defined median, and also you can use undefined media. So the undefined median, so that'll be like a L'Oreal Bretagne LB culture, and which is commonly used in the laboratory to prepare the cell, to prepare to grow the bacteria. All right, and the maximum density is about the 2 to 3 times 10 to the 9. All right, and and then to measure the amount of a cell in the culture. Okay, yesterday I think I briefly described this. You can use the indirect method. So this is the indirect method. You put the bacteria culture in a cuvette, and then bring this cuvette to the spectrophotometer and read the optical density at the wavelength 600 nanometer. And every one optical density, right? Every one, at the 600 nanometer, that will be equal to 0.8 times 10 to the 9 cell per milliliter. Okay, any question? Nancy. Still missing 3 people. Two more. Where are the other 2 people? Muhammad, I did not ask you. Lilac, Mohan, you did not respond. Okay, so this is very important that will be in the quiz, will be in the exam. Okay, you have to know how to calculate the amount of the cell if the OD reading is given. All right, any question? Is, for example, if an overnight culture, then I dilute it 10 times, and then I put it, and then if I dilute the overnight culture, and then I put it into the cuvee, and then to read the OD, and then if the OD is 0.5, then I will ask you what's the amount of cell in the culture. So you have to know how to calculate the bacteria quantification. Okay, and yesterday you also performed the Labuster, that's a serial dilution, and that would be one of the direct methods to determine the amount of cell. Okay, so that's the optical density in direct method to determine the amount of cell. And then of course the first step after you grow the cell overnight, then the first step you need to do is to harvest the cell. To harvest the cell, then you have to use the centrifugation. And this is just the diagram to show you the centrifugation. Then you can collect the cell pellet. And then after you prepare the cell pellet, collect the cell pellet, then you have to disrupt the cell so that cell will be able to release the cell components, which include in the DNA inside a cell. And of course, you can use a physical method to break the cell. That would be the mechanical force. So that would be like the sonicator that we normally use in the cell, in the laboratory sonicator that you can break the cell. All right, that's a physical method. Or you can use the chemical method, that involve the chemical reagents, right? And those chemical reagents, for example, here you can use the lysozyme. Lysozyme can weaken the cell wall, and then also the EDTA, all right? And then the detergent, okay, this part is tricky, so you have to pay attention. So here I put the SDS. So the SDS is the detergent, the ionic detergent. Okay, the ionic detergent. If you use SDS, normally the SDS will break the genomic DNA. Okay, break the genomic DNA, so you become the linear. But if you are going to prepare the plasmid DNA from the bacteria cell, then it depend on the downstream, the plasmid DNA isolation method. Then you have to decide whether to use the SDS or other non-ionic detergent. Okay. So and here we can use SDS, all right? And then after you use this chemical reagent, okay, then you can disrupt the cell wall and then release the cell extract, all right? And then after the centrifugation, then you leave the DNA, RNA, protein in the supernatant. Then you can further purify the DNA from the supernatant. Okay, and the chemical reagents. So here, for the lysozyme, that's the enzyme that can digest the cell wall. All right, and for the fungi, like the yeast, then you have to use a different kind of enzymes called lyticase. All right, so for the Saccharomyces cerevisiae, you have to use the lyticase so that they can break the cell wall. And sometimes you can the CTAB, and this CTAB can also be used when you try to isolate the DNA from the plant, all right, from the plant, the CTAB. And the Chelax 100 is a chelating resin, all right, and has a high affinity for the polyvalent metal ions. So can also be used to isolate the fungi DNA quickly and efficiently. So it is frequently used to release the DNA from cell by a boiling treatment, at the same time protecting the DNA from the boiling effect with the resin beads. Okay, so typically, if you use the candida, so the candida suspension from the sample is extracted by adding 5% of the Chelax 100 resin, and then you incubate the mixture at 90 degrees for 30 minutes, and then after that you centrifuge the sample, and then you incubate again at 90 degrees for 15 minutes, and then later on you collect the supernatant after the centrifugation. So the Chelax extraction method is quick to obtain DNA from the spore, like those are fungi spore. Okay, so that's another method that normally people use. And then after that, as I mentioned, after the centrifugation, you separate those DNA, RNA, protein from the cell debris. And the next step, right, the next step is to purify the DNA from this supernatant. And then to purify the supernatant, then we have three different methods. One is organic method, use the organic chemicals like phenylchloroform to isolate, purify the DNA from the supernatant. Or you can use organic method. Basically, you just precipitate the DNA. All right, use a salt to precipitate the DNA in the presence of the ethanol. Or you can do the solid phase. All right, and then so basically the supernatant goes through the column, and then the column will bind the DNA, and later on you can elute the DNA, right? So this is the method like using the phenylchloroform. So the idea is the phenol will be able to precipitate a protein. Okay, if you mix the supernatant with the phenol, and then apply the centrifugation. After the centrifugation, then you can see if you mix with the phenol. After the centrifugation, the protein will be precipitated in the interface between the aqueous layer, which contains DNA and RNA, and the phenol here, right? Phenol being the bottom, right? So that's the phenol. If you apply the phenol and then you mix well and then apply the centrifugation, then you precipitate the DNA, precipitate the protein, and then you will remove this supernatant, the aqueous layer, the supernatant here, aqueous layer, and then you mix with the chloroform, and then mix it with chloroform and then apply the centrifugation. So after the centrifugation, basically the chloroform, the function of the chloroform is to remove all the residual phenol from the DNA RNA here. Okay, so that's why you have to use the phenol and chloroform, so that the aqueous layer will be pure, only DNA RNA. And then to remove the RNA, then you can use the RNase, all right? RNase, which can digest RNA and leave the DNA behind. So that's basically the organic extraction. And in the meantime, you can also use the proteinase K, all right, during the phenylchloroform extraction, because proteinase K can help to digest the protein in the sample. So that's the organic method to purify the DNA. For the inorganic technique, right? So the inorganic method involves the incubation of a nuclei with the proteinase K, all right? The proteinase K basically will digest all the protein at a 65 degree for 2 hours, okay? Because the proteinase K is more active and denature the protein, and also after prolonged incubation at 65 degree can be auto inactivated, all right? So then following incubation for more than 2 hours with the proteinase K in the low-salt buffer, the extracted DNA can be used directly for diagnostic analysis without any additional purification, all right? So basically that's proteinase K. And then after that, you can do this, you can saturate with the sodium chloride, and then you can in the presence of ethanol and cold temperature, so you can precipitate the DNA, all right? You can precipitate the DNA. That's inorganic DNA isolation. And then for the solid phase DNA isolation, that this solid phase, basically you have the column, that a lot of these can be found in a commercial extraction kit in the market. So these allow a quick and efficient purification, okay, compared to the previous two methods. So basically the solid phase system will absorb the nucleic acid in the extraction process depending on the pH and the salt content of the buffer, right? The absorption process based on the principle that the hydrogen bonding interaction with the hydrophilic matrix under the chaotropic conditions, and the ionic exchange under the aqueous conditions through an ion exchanger. That's because DNA is negatively charged, all right? So you have to use if you use the ionic exchange, then you have to use an ion exchanger, or you can use the affinity size exclusion mechanism to purify the DNA. And the commonly seen the spin column, that's quite commonly seen. So basically you have these pellet bacteria, and we suspend the lysis buffer, And after lysis buffer, if you apply the centrifugation, and then you have the supernatant, and then run the supernatant through the column, then the DNA will bind to the column, all right? And later on, you can elute the DNA. Okay, that's the solid phase DNA isolation. And this is to show you the affinity extraction of the DNA. Oh, actually, this is the wrong. This is the not a correct explanation, the correct picture for the extraction of the DNA from the column, because some of the column they have a magnetic separation. Okay, so through the magnetic separation, basically the magnetic carrier with the immobilized affinity ligand prepared from the biopolymer, they have affinity for the specific target nucleic acid, and then you can use that to separate the DNA from all other component in the cell X-ray. Okay, and this is to show the anion exchanger, right? So you use the anion exchange resin to attract the negative charged DNA and then separate the DNA from other materials. So that's the anion exchange extraction. And then after you extract the DNA, then you can precipitate the DNA to concentrate the DNA. So in order to concentrate the DNA, you need to use these ethanol, salt and ethanol to precipitate the DNA. And we normally call this ethanol precipitation because you need to have the 0.3 mole of the salt and plus 2 volume of the ethanol in the presence of a negative 20 degrees Celsius, and then the DNA will be precipitated. And so after that, then you can concentrate your DNA. And to determine the DNA concentration, you have to use the UV light, the UV absorbance at the 260 nanometer. And every one absorbance will be equal to a 50 microgram of a double-stranded DNA. All right. If it's single-stranded DNA, then every 1A260 will be a 40 microgram. So that's the how to determine the DNA concentration. Any question? Brooke. Neil, it's in the textbook, yes. But not all of them in the textbook. We're still missing some people.
It's kind of distracting when you making us to call it. Like for example, me, like I'm trying to take a note when you ask me and just like forgetting my thought.
Sorry, what did you say, Nadia?
It's kind of distracting when you make us call us, like asking us questions to say no, yes, just for like, oh, if we are here.
Yeah, of course, I have to make sure you understand everything I say. So that's why I have to ask if you have any question.
I'm
I
trying
need
to take notes, and when he asked me, like, I just forgetting my thought, just like need to
Oh, I see.
say no, yes, be in the chat.
Okay. Rodana. Rodina, do you have any question? Good. So Neil, so what I just present, yes, that's the class material in the content section. You have to check the Blackboard, but this material I just posted right before the class, okay? Right before the class. So we just finished the standard procedure that to prepare the DNA from the bacteria cells. Okay, so for the plant cell, basically you use CTAB and also some fungi that you also use the CTAB. Okay, you can use these in the cell extract, the plant cell extract, because you can precipitate the nucleic acid. Okay, by using the CTAB. And so that leave the carbohydrate, protein, other contaminant, the supernatant. So here's the, for example, this is cell extract. Then you mix with CTAB, and then because they can form a complex with the nucleic acid after the centrifugation. So you basically precipitate the nucleic acid CTAB complex, all right? And then you remove the discarded supernatant, and then resuspend this pellet, all right, in the 1 molar sodium chloride. Okay. Then that will break this nucleic acid and CTAB complex, and then that will leave the nucleic acid in the supernatant. All right. That will leave the nucleic acid in the supernatant. And then you can use the same technique here to concentrate your DNA, or you can go through this phenylchlorophyll extraction, all right, to purify this DNA here. Okay, so everything will be the same after this step, because this can be the phenylchlorophyll extraction plus ethanol precipitation. Just the treatment beforehand will be different. Okay, and then another technique is the guanidine cyanide. Okay, that can basically the guanidine cyanide can denature and dissolve all the biochemicals other than the nucleic acid, then can be used to release the DNA from any type of cell or tissues. Okay, and also in the presence of the guanidine cyanide, DNA can bind to the silica particle strongly. Okay, so when you go through this column, and then they can bind here strongly. Later on, then you use the water or TE to elute the DNA. So that's a guanidine cyanide. So these are all the techniques that you can purify the DNA from the cell. And the next, we're going to focus on the plasmid DNA. So the plasmid DNA, as we discussed in the past 2 days, if you introduce a plasmid DNA into the cell, and then you put the cell in the selected media, and for all those colonies you observe, that just prove the recombinant phenotype. And then to prove the recombinant genotype, then you will need to purify the plasmid DNA from bacterial cells or the bacteriophage, right? And and remember, when you try to purify the plasmid DNA, in the meantime, there's also genomic DNA, bacterial genomic DNA in the cell. So the purpose of this technique is to separate the plasmid DNA from the genomic DNA. okay? And then there are two different type of methods that you can use to separate the plasmid DNA from the chromosomal DNA. So one is based on the size. That's because the plasmid DNA is always like 8% or less of the chromosomal DNA. And the second method is based on the conformation. And the conformation is because the plasmid DNA usually is a supercoil, okay? Supercoil because it's circular, so normally you will wrap into this supercoil DNA, okay? So based on the size of conformation, then we will be able to separate the plasmid DNA from the chromosomal DNA, all right? And now, based on the size, remember in the beginning that when I mentioned that to disrupt the cell, we can use SDS, but the SDS is ion detergent. And then I also say depend on the method that you want to use to isolate the plasmid DNA. Then you have to use different kind of detergent. So if we're going to use the size fractionation to separate the plasmid DNA from the genomic DNA, then we have to use the non-ionic detergent. That means you cannot use SDS. You have to use the Triton S100. The Triton S100, because the Triton S100 will not break the genomic DNA. That will keep the genomic DNA maintained intact. So that's the point, because we want to make sure the chromosomal DNA is not broken. Okay, so you have to use non-ionic detergent, and that would be the Triton S100. So in this process, so it's the same. Use lysozyme EDTA. In the meantime, you have 25% of the sucrose, so that you can produce this spheroplast. And then spheroplast, then you use the Triton S100, and then to release all the cell X-ray. And then the cell X-ray contains both plasmid DNA and also the intact genomic DNA. And now you can apply the centrifugation. After the centrifugation, you will precipitate the cell debris and also the large DNA molecule, which will be the genomic DNA, will be in the bottom. And that will leave the plasmid DNA in the supernatant. And then after this, you can remove the supernatant. This is clear lysate contain the plasmid DNA. You can remove this supernatant and then perform the phenylchloroform extraction. Or you can do the inorganic method to sort out the genomic plasmid DNA, or to go through the spin column to collect the plasmid DNA. So that would be the size fractionation. And then also you can use based on the conformation to separate the plasmid DNA from the genomic DNA. And then there are two different techniques that you can use. You can use the alkaline denaturation, all right, alkaline denaturation, or you can use the acidity bromide cesium chloride density gradient centrifugation. You can use these two different methods that to isolate, to separate the plasmid DNA from the genomic DNA. So the figure here basically is the acidity bromide cesium chloride density gradient centrifugation. Okay, so basically you mix the cell extract with the cesium chloride, right? And then you have increase in the cesium chloride concentration after the centrifugation. Then the DNA, all right, based on because different density, then based on different density, then you can separate the DNA from protein RNA. And so that's the general idea. So now if you have the plasmid DNA and the chromosomal DNA, okay, then after you break the cell and release the cell X-ray, then you use the cesium chloride, ethylene bromide cesium chloride density gradient centrifugation. After the centrifugation, then you can separate this linear DNA and the supercoiled DNA. The supercoiled DNA will be the plasmid DNA. The linear DNA, that would be the genomic DNA. And then because this procedure, you add ethylene bromide, ethylene bromide in the solution, so you'll be able to see the DNA band. Then you can remove this lower band, which will be the supercoiled DNA. That will be the plasmid DNA. After you use the needle and then to remove it, and then you are checked with the butanol, right? You check with the butanol so that you can separate the ethylene bromide from the DNA. So the ethylene bromide will be in organic phase, and then the bottom one will be the DNA in the aqueous phase. And then you can remove this supernatant, the aqueous phase, and then to do a dialysis to remove the cesium chloride. And then after that, then you can do the phenylchlorophyll extraction and the ethanol precipitation to concentrate the DNA. Okay, that's the procedure to purify the plasmid DNA using ethylene bromide cesium chloride density gradient centrifugation. And then for the alkaline denaturation, which is the most commonly used technique. So basically, the idea is you use this SDS, so you and lysozyme, and then to PDTA, you break the cell, you get the cell X-ray, and then at this time, because you use the SDS, so all the genomic DNA will be broken down to the linear DNA. All right. In the meantime, the supercoiled DNA still maintain the supercoil. That's plasmid DNA. So after you collect the cell X-ray, the first step you need to do is to bring the pH to 12. So to bring the pH to 12, then you have to add a sodium hydroxide to the cell X-ray. Then bring the pH 12. At this time, this linear DNA will be denatured. All right, denatured becomes single-stranded, and then the plasmid DNA remain intact. And then after this, then you bring the pH back to the neutral, so that those linear DNA will renature imperfectly. After they renature imperfectly, then you can apply the centrifugation so that you leave the supercoiled DNA, now be the plasmid DNA in the supernatant. All right, and then you pellet all the debris and also the linear DNA in the bottom, so that you can remove this supercoil, and then this supercoil is subject to the phenylchlorophyll extraction and the ethanol precipitation, and then you can get the pure plasmid DNA. All right, any question? Rodena, any question? Rodena, any question? Muhammad, just a second. Rodena. Okay. So the plasmid DNA are circular DNA, and then because it's circular in the natural environment, you will form the supercoil, all right, and supercoil, and that's because the physical difference. Yes, that's a physical difference. The conformational difference is a physical difference between the plasmid DNA and also the genomic DNA. All right. So if you use the SDS will break those genomic DNA into the linear DNA. If you use the Triton S100, then that will maintain the genomic DNA intact. So that's based on how you're going to proceed to isolate the plasmid DNA, then you have to choose different detergent to process your cell. Okay, if you're going to isolate the plasmid DNA based on the conformation, then you have to use the SDS. If you're going to separate the plasmid DNA from genomic DNA based on the size, then you have to use the non-ionic detergent, so like a Triton S100. Okay, so that's the process to isolate the plasmid DNA. Okay, and then the next few slides just to show you to isolate the DNA from the bacteriophage, because remember in the beginning that when we discuss the cloning vector, we have a vector based on the E. coli, and then you're going to introduce those vector into the bacterial cell. Also, we have the vector based on the bacteriophage. So that means we're going to introduce those vector into the bacteriophage, okay? And so that we use the bacteriophage to infect the bacterial cell so that they can reproduce the DNA. And then for the bacteria, prepare the bacteriophage DNA. So basically, again, you grow the bacterial cell, and then you add the bacteriophage to infect the bacterial cell, and then after that, then you apply the centrifugation. All right. After the centrifugation, you have the phage suspension. Then we can process this phage suspension to get those DNA that we introduce into the phage. Okay. So this is just to show you that to grow the bacterial cell, and then you introduce the phage, so that the phage will infect the bacterial cell and use the bacterial cell to reproduce the phage. Okay, so and then if the culture density is too low, then you add the lambda inoculant, and then so the cell will be rapidly lysed, and then you have a very lower phage titer. If the bacterial culture density is too high, then the culture is never completely lysed, and also you get a low phage titer. And then you have to do some experiment to determine the optimum condition, so that the amount of the phage you add to the bacterial culture will make sure that the phage have enough time to infect all the cell, and then reproduce, and then you get the maximum product. Okay, that's a high phage titer. So that would be some instruction that when you use the bacterial phage to multiply the DNA. Okay. And then for those, so after you infect the cell with the phage, so and then you apply the centrifugation here, so you get the phage suspension. All right. After you get a phage suspension here, Then you can add a PEG, which is polyethylene glycol, and the sodium chloride, and then you can precipitate, all right? Precipitate the fudge, all right? Precipitate fudge. And then you can resuspend the pellet. Resuspend the pellet, and then you can do the phenylchlorophene extraction. All right, phenylchlorophene extraction of these resuspended pellet, so that you can purify the DNA. And main thing is the PEG, all right, polyethylene glycol and sodium chloride to precipitate the fudge particle. And then for M13, again, so you have the culture of infected cell plus centrifugation. So you have this fudge particle suspension, then you remove this fudge particle suspension, and then you add PEG, sodium chloride, you precipitate the fudge, and then you discard the supernatant, resuspend the fudge in a buffer, then you can use the phenylchlorophene extraction. All right, this is phenylchlorophene extraction to get the pure DNA. So these are the treatment for those fudge DNA. Okay. And now we're going to watch a video about a DNA purification. Okay, quickly. It's okay, then we'll do this one here.
Before beginning any protocol, you want to make sure that your workspace has a proper flow to avoid contamination of samples or reagents. This example is set up for a right-handed student. Tips and pipetters are on the left.
Pipette is a very important tool, all right, because in the molecular lab, that for everything that you process all the sample, you always need to use a pipette. So that we have one lobster simulation is to show you how to use the micropipette. Okay, this is extremely important because the micropipette, if you don't know how to use it or you use it incorrectly, then you will not be able to get the result. Okay.
Racks and samples are in the middle, and the discard bucket is on the right. As your hand moves through the flow, it never unnecessarily crosses over the sample again. This video begins after students have spit into the DNA Genotek collection kits, capped the tubes to release the buffer, and mixed the solution by inversion. At this point, sample tubes should not be labeled to preserve student anonymity during the genotyping process. Using the P1000 micropipetter, 500 microliters is transferred from each spit collection tube into a new microcentrifuge tube. When pipetting large volumes, draw up the solution slowly to avoid bubbles or contaminating the micropipetter.
Have you all used the micropipette before? Very good. So when you use a pipette, it's always very important. You have to have the tip in the place, and then you depress the plunger. All right, depress the plunger to the first stop, which is very gentle, very soft stop, and then you insert the tip into the solution. and then you slowly release the plunger, and in the meantime, the pipette tip will draw the solution into the tip here. All right, and then when you're going to release the solution inside the tip, you have to depress the plunger to the first stop, and of course the tip has to touch the wall, the wall of the tube. All right, and then you go to the button and then touch the wall, and then slowly depress the plunger to release the solution until the first stop, and then you quickly depress the plunger again to the second stop, and then in the meantime you quickly remove the micropipette so that you can dispel every single drop inside the pipette tip into the tube. All right.
Once the solution is transferred, close the spit tube lid and cap the sample. Repeat this process for all remaining samples. Prior to beginning this protocol, the heat block should be dialed to 50 degrees and turned on by flipping the switch. If it's heating, the light will glow red. Place samples in the heat block. This incubation must proceed for at least 90 minutes, but can also be left overnight. Tubes containing 20 microliters of DNA Genotek's proprietary purifying solution, PrepIt, will be provided.
The heat block is just to break the cell, okay?
Once the samples have been incubated, you will transfer the entire volume to a tube containing the purifying solution.
Yeah.
You may notice that the solution in the tube becomes cloudy. This is normal. Mix the solution with a vortex.
Yeah, the vortex is a very commonly seen
To turn on
equipment
the vortex,
because you can use the vortex to mix everything. So, for example, when we add the phenol chloroform into the cell X-ray, you have to use the vortex just to make sure they can mix completely.
press the switch down to touch and turn the knob clockwise to ensure maximum speed. Hold one tube at a time and press the top of the black pad to initiate vibration.
This is what we normally do in the lab, use the vortex to mix the
Repeat
sample.
this process for all remaining samples.
Yeah, when the campus will reopen again, all of you are welcome to come to my laboratory. I can give you a lab tour to show you all the equipment that we use. So for even this vortex, heat blocks, everything. Okay. And if you want to do the DNA isolation, yeah, you can also do it in my lab too. But we have to wait until the campus reopen, which most likely will be in the fall semester.
Embed all samples in ice for 10 minutes. Impurities in the solution will be removed by centrifugation. After flipping the switch in the back of the microcentrifuge, press the open button. Remove the metal lid inside by pulling on the knob. Make sure that the tubes are labeled either with letters or numbers before loading them into the centrifuge. Load samples with hinges facing outward.
Yeah, that's very important. The hinge face outward so that all the precipitate will be in the certain direction.
Have equal volumes. Replace the metal lid and press it until it clicks securely onto the rotor. Close the lid to the microcentrifuge gently. Change the centrifugation time by pressing the arrows to the desired time, in this case, 5 minutes. Press the arrows by the speed to navigate to the proper RPM.
There's a speed, that's a RPM, that's a run per minute, so it's multiply 1,000, so that's a 14K, all right? That's normally what you can do for the microcentrifuge, all right?
In this case, 14.5 thousand RPM is the max this centrifuge can perform and is close enough to our desired speed. Press the start button. The centrifuge will begin to spin. It is suggested that you watch the centrifuge until it reaches maximum speed to ensure that no violent vibrations, which may mean that the tubes are unbalanced or the internal lid has come loose. If this occurs, press the stop button immediately.
And when you place the tube inside the microcentrifuge, and it's very important that you have to balance the centrifuge, all right? Balance centrifuge. That means if you put the one tube at one side, and then across the center, in the other side, the opposite side, you have to put another tube, and so that you can balance the rotor.
As the spin is going, obtain 4 new tubes and label them. When the spin is complete, the lid to the centrifuge will pop open. Remove the internal lid and remove the samples gently to avoid disturbing the pellet. Place tubes of the same sample in line with each other to ease the transfer of the solution. After centrifugation, you should be able to see a white pellet that has collected on the bottom part of the tube on the side of the hinge. This pellet contains the impurities in the solution. The DNA is in the supernatant. Using the P1000, transfer 400 microliters of supernatant, which contains the DNA, to the new tube.
So after all the treatment, now you transfer the supernatant. That includes the DNA.
Place the tip on the opposite side of the tube from the pellet and draw up slowly to avoid disturbing it. Repeat this process for the remaining samples.
So after that, you collect these supernatant, all right? Collect the supernatant, and then after that collection of supernatant, then you will have 3 different choice, all right? 3 different methods you can proceed to purify the DNA. So one is the organic extraction. Number 2 is inorganic extraction. Number 3 goes through the spin column. Those are solid phase, and to purify the DNA. Okay. And then let's go to the step here. The part 2. All right. So now they have the part 2.
precipitate the DNA from solution by adding an equal volume of 100% ethanol to each.
So basically, in this protocol, they use this the inorganic method. So they add the ethanol and salt directly in the cold temperature, so that they can precipitate the DNA. All right. So that's the one method they choose. And of course, if you want even pure DNA, it's better. It's the best is to go through the phenylchlorophene extraction. And the problem some people worry is that because of phenylchlorophene, those organic chemical can cause a contaminant of the environment. So people try not to use the phenylchlorophene. But personally, if you want a pure DNA, and you should use the phenylchlorophene extraction. Okay.
Sample. And mix by inversion 10 times. The solution may become cloudy. This is normal. After incubating at room temperature for 5 minutes, place the samples in the microcentrifuge with the hinges facing outward. Remember to balance your tubes.
And then, as you can see, she placed the tube in opposite direction so they can balance the rotor. Okay. And also, she used these 5-10 minutes in the room temperature. But normally, you have to, if you want to get the maximum recovery, you have to put into the cold temperature. All right, the cold temperature that can get the maximum recovery.
Spin the samples for 2 minutes. Remove the tubes from the centrifuge gently, being mindful not to disturb the pellet which now contains your DNA.
Yeah, because you use ethanol precipitation to precipitate the DNA. So you
visually inspect for a pellet at the bottom of the tube on the side of the hinge. A pellet is not always visible, so you can choose to centrifuge again or proceed as normal. In this example, a pellet is only seen in samples C and D, but all samples are carried through the process.
Yeah, the pellet normally is very small and barely to see that, okay?
Using a P1000, slowly remove and discard approximately 800 microliters of soup.
And for this step, basically you do not need to use the micropipette to remove the supernatant. You can just discard the supernatant by just inverse the tube and then open the cap and then just throw those supernatant directly, because the pellet right after the centrifugation, you have to perform that to discard the supernatant immediately. The pellet will still maintain, stick to the tube wall, so you will not lose the DNA. Just open the cap and then invert the tube and then throw out the supernatant. That's the easy and quick way. No need to use the microcentrifuge, and no need to use a micropipette. The DNA should be, the precipitate should be on the side of the hinge.
Place the tip at the bottom of the tube on the opposite side of the pellet to avoid disturbing it. It is not important at this point that all solution is removed. Just get as much as possible. Expel the tip with the solution inside into the discard bucket. Repeat this process for all remaining samples.
The reason they don't need to remove all the supernatant is because the next step, you can wash the pellet again, or you can air dry.
Inspect the tube to see if this pellet is still present. If it has gone missing, make a note of that. Clean the DNA pellet by adding 250 microliters of 70% ethanol to each sample.
So this is to wash the DNA pellet. All right. Remember, before the precipitation, to try to precipitate the DNA, you use the 100% ethanol, and then to wash the DNA pellet, you use the 70% ethanol.
Do not mix the solution, as it is imperative that the pellet is not disturbed. Repeat this process for all remaining samples. Place the samples in the centrifuge, being mindful of balance, and spin for 2 minutes. Remove the tubes from the centrifuge carefully. In this step, the pellet still contains the DNA, and it is important to remove as much supernatant as possible without disturbing the pellet. So remove and discard the supernatant using the P200 for precision.
Oh, sorry. How do I move the? Just a second. So the last step basically to remove those supernatant, all right, and then after that you have to use either air dry, all right, or the speed vac dry to remove all the supernatant, because the DNA is in the pellet. So make sure you have to make it dry completely before you can dissolve the DNA in the trace EDTA solution so that you can keep the DNA for a longer time. And at that time, I tried to, I tried to stop it because they show you the micropipette with the yellow color on the top of the plunger, all right? And it showed a 200. And then the other one is a blue tap, is 1,000. So the difference between these two micropipettes, so basically the blue tap for 1,000, that means the maximum volume is 1,000 microliter. Okay, and and then the other one, the micropipette with the yellow colored tap, that's the 200 microliter, and they use a different kind of the tips to remove or to draw the solution. Okay, so that's just to show you the difference between those two micropipettes. Okay, so that would be today's lecture. Any question?